Hello, my name is Lan-Nhi Phung, and my talk today is Comparative Analyses of Vocalization and Genomic Divergence in Nashville Warbler. I will start with my evolutionary interest, which is the process of biological speciation through reproductive isolation. I'm specifically interested in the role of vocalization as a trait and reproductive barrier, and how genomic studies and biogeography can aid this understanding. Genomic analyses can tremendously help clarifying previously confusing species relationships on a systemic level, such as this revised phylogeny of the wood warblers based on mitochondrial DNA and nuclear DNA data in 2010. This is an example on how studying the entire genome instead of already known individual genes can help gain a more objective and zoom out view of how selection works. In terms of trait assessment in birds, the study of reproductive isolation had been prominently with plumage. This Iberia seed eater study highlighted this using genomic data to pinpoint genes and their function that affects plumage-related traits, which in turn affect the gene flow of species in symmetry. Regarding biogeography component, this golden wing and blue wing warbler study highlighted geographical variation of plumage-related traits in the context of hybrid zones. Hybridization signals weakened reproductive barriers between two populations. Therefore, the divergence of genomic components that related to plumage could tell us about traits that are significant reproductive barriers. So combining these components, treating vocalization as a reproductive barrier and examining its relationship with geographical variation and genomic variation, there are much left to be understood about the role of vocalization in evolution and speciation. And moreover, while the study of sympatric and hybridizing species are rich, expanding our understanding of divergence in allopatric species would also be crucial in the understanding of the speciation process. So with that said, Nashville Warbler is a perfect candidate for this kind of study for two primary reasons. First, it consists of two subspecies, Leothlybis ruficapilla, Ridgway eye, and Ruficapilla ruficapilla, who are allopatric and therefore have no hybrid or contact zones. Second, the most recent genetic work on this species was in 2004, which examined mitochondrial DNA to determine differentiation of haplotypes, and the two subspecies correlate with two haplogroups. In our study, we will further explore this separation by using nuclear DNA with a more advanced sequencing method. Moreover, the two subspecies have indistinguishable plumage and songs that are quite distinguishable to the ears as well as on spectrograms, but were never analyzed systematically. Here are the examples of the two songs, the western subspecies and the eastern subspecies. So in the vocalization analysis, we examined 24 acoustic characteristics. Song recordings were acquired via the Macaulay library and analyzed using Raven Pro. We purposefully chose recordings all across the subspecies ranges for adequate geographical representation. We used a couple of analyses to see if the song characteristics could characterize the individuals. First, we used principal component analysis to specifically find out which characteristics are the main drivers for the differences, if there's any. And second, we use a blind clustering method called partitioning around medoid, or PAM, which determines group membership of each individual without a priori input. With PCA, our results showed that the individuals of two subspecies separate gradually along PC1, which explains 21.26% of the variation between individual songs. The clusters are not distinct, and there are some overlaps, which we interpreted as strong individual variation within each subspecies. The square loading of PCA showed that some characteristics seems to be more important than others, especially the number of repetition of syllable B and minimum frequency of syllable B, which have the largest loadings in PC1. Overall, it seems that characteristics of the latter part of the songs are what makes the songs more diverged. With the PAM analysis, our result also showed two diagnosable clusters with some overlaps. The bigger points in the middle are the medoid, and the closer an individual is to the medoid, the more likely that it belongs to the cluster. So cluster 1 is predominantly Western individuals, Cluster 2 is mostly eastern. You can see that some individuals were grouped in cluster 2, even though they're closer to cluster 1's meadowoid. Again, this is potentially because the songs are individually varied enough from their typical subspecies songs that made them sort of intermediate and harder to put into either cluster. And as we put the clusters onto a geographical map, we see this result more clearly, that there's a geographical effect. So the difference in songs is associated with the geographical variation of the two subspecies. Obviously, the overlaps had also shown through here, but the West still predominantly consists of Western songs and the East of Eastern songs. Additionally, we found no dialectical differentiation within the subspecies, though there seems to be lots of individual variations. Onto the genomic analyses. So for this part, we had 24 DNA samples taken from blood or tissue sequenced with Illumina NextSeq, aligned to the new yellow rump warbler reference genome and analyzed using a bioinformatic pipeline that results in a population level output called the fixation index, or FST. Simply put, FST is a pairwise comparison between two populations, which shows how each single nucleotide polymorphism, or SNP, on one set of data is different from the SNP at the same location on another. I won't go into details of our bioinformatic pipeline here for the sake of time, but please ask questions if you have them. So the FST goes from 0 to 1. Closer to 0 means less differentiation, closer to 1 means more. Genome-wide, our results showed an FST of 0.087, which means in general, the two subspecies are very similar. However, the genomic PCA here showed that individuals sorted themselves into their subspecies. Here you're seeing 12 individuals of each. There's no admixed individuals, meaning there's no contemporary hybrids within our sample. So there's some level of reproductive isolation, but where are the differentiation on the genome specifically if they're not affected via the global FST? Here, I'm visualizing FST of the entire genome as a Manhattan plot. So the y-axis goes from 0 to 1, each peak is a SNP. What we're seeing here is that though there's an overall low background of divergence, there's also dozens of high peaks that show the position of the most differentiable SNPs. Zooming into one of the chromosomes, you can see the magnitude of the peaks comparing to the background that are mostly between 0 and 0.2, while the peaks are about 0.7. Interestingly, in the Parulidae family, there are other closely related species and subspecies pairs that has varying degrees of divergence that gave evidence for species boundaries. Here I'm showing chromosome 1 of some previously compared pairs. So the schematics on top of each plot represent how much divergence there are accordingly to the plot. So graces and black-throated gray warblers are highly diverged across the board. Golden-winged and blue-winged warblers diverge very little and in fact actively hybridize. The divergence of the Nashvilles is the closest to the divergence between the two yellow rump warblers, both of whom background divergence are low, but highly diverge at similar loci. So to summarize, using the Nashville Warbler subspecies pair, we found that the two subspecies are diverged in both vocalization and the genomes, but more so in the genomes. For the vocalization, we found that though there are individual variations, the songs are diagnosable and especially different in the second part. For the genome, we saw a low overall divergence with many high divergence SNPs. This pattern is very similar to that of the Audubon and Murdoch's warblers, members of the yellow rump warbler complex that was shown repeatedly to be highly diverged, where there are fixed SNPs at important loci and overall limited gene flow. This is evidence that the two natural subspecies might have the same trajectory, and further study on this topic is worth pursuing to re-examine their species status. In the next step, we will first address some of the limitations of our current study. So for the genomic analysis, our Eastern samples were strictly blood samples from the Adirondack, and the Western samples were tissues that were kindly loaned to us by the Bird Museum. With our promising result, we intend to broaden our sampling to the rest of the species range to assess the pattern more accurately. Moreover, our FST analysis presented only a summary surface level understanding of the genomes with a low coverage data set. Therefore, it will be beneficial to apply deeper analyses such as a demographic history modeling to quantify the gene flow specifically. This was implemented reliably as evidence for examining species boundaries, as was done in the study of the similarly allopatric willet. We could also do habitat modeling to understand the effect of climate niche differences between the two ranges. Lastly, we would like to address the behavioral aspect that is lacking, which is to do a reciprocal playback experiment to assess song recognition and pre-mating isolation. These components will hopefully elucidate the biological relevance of the initial findings we found in this current study. And with that, I would like to thank members of the TAVES lab for helping me with both wet and dry lab components of the study, and also just generally being supportive. I'd also like to thank the biology department at Penn State and the Huck Institutes for their funding support. Special thank you to the Bird Museum for their generous tissue loan, and the Macaulay Library and their contributors for providing me with song recordings. My work would not have happened without you all. And I will gladly take questions. I'm also on Twitter at Lan-Nhi Phung if you would like to contact me afterwards. Thank you for your attention.